Guys, we are looking at Staphylococcus. Staphylococcus antigenic structure. If they ask you, 4 things you should write. Capsule, Peptidoglycan, Teichoic acid, protein A. Fine? Can you say it, all of you? You say capsule, Peptidoglycan, Teichoic acid and protein A. Okay? Now, some strains possess capsule. Okay? So, what are the functions of capsule? To maintain the shape of the cell. It has antigenic function. It inhibits phagocytosis. It makes it more virulent. All that, right? So, you can write that here. Capsule strain become more virulent. They are made up of polysaccharide. What are they made up of? Made up of polysaccharide. Okay. There is a antigenic structure diagram here for Staphylococcus aureus. This is the cytoplasm inside. Outside you have a capsule. Then you have the cell wall, cell membrane. See, cytoplasm, cell membrane, cell wall, and capsule. This is not a difficult diagram at all. Okay. In cell wall, they have put 2 components: peptidoglycan, teichoic acid, and protein is written here. Guys, don't worry so much. Here you have inside cytoplasm, outside that cell membrane, outside to that you have cell wall, and outside to cell wall you have capsule. Okay. Moving on to the next one, you have peptidoglycan. Peptidoglycan is going to be antigenic and it will produce inflammatory cytokines. It provides rigidity to cell wall. Fine. Teichoic acid. Teichoic acid is a major antigenic determinant. Please note, it is the major antigenic determinant. Major antigenic determinant, that is teichoic acid. It is the group specific ribitol teichoic acid. Okay. It is ribitol teichoic acid to be more specific. It facilitates adhesion of the cocci to the host. This is interesting. It is going to help in the adhesion of the cocci to the host. It protects it from the complement mediated opsonization. So this is very, very interesting. So it is going to make the bacteria more virulent. It is going to protect it against opsonization. Who is going to do opsonization? Complement mediated here. And it is associated with peptidoglycan. Always teichoic acid is associated with peptidoglycan. And it is absent in staph epidermidis. Why am I saying that every time? Staph epidermidis. Okay. And the latter contains glycerol teichoic acid. Staph epidermidis has glycerol teichoic acid. Staph aureus has ribitol teichoic acid. Let me write that here for you. Staph aureus has ribitol teichoic acid and staph, and staph epidermidis has glycerol teichoic acid. Fine. Coming on to the last one, that's protein A. Protein A is a component of the cell wall again. Even peptidoglycan, teichoic acid, all these are cell wall components. Protein A is also cell wall component. What you need to know is in the staph aureus, that is especially Covan 1 stain, it is present a lot in the cell wall. Okay. Again, you should know it is antiphagocytic and anti-complementary. So, it will resist phagocytosis, it will resist opsonization. Okay. And it is going to bind to the Fc portion of IgG. How is this going to help? Normally, where should it bind? Normally, where does the host want the IgG to bind? See, it can block phagocytosis by this. So, it blocks phagocytosis because it binds to IgG in the Fc portion. So, you can see here, this is the FAB portion and this is the FC portion. FAB portion is where the normal binding happens. Okay. But Staph is going to bind in the Fc portion. So see the name, okay, FAB, FAB is fragment, antigen binding. This is the other fragment, that is crystallizable region. So normal antigen binding has to happen here, but Staph is going to bind to the Fc region. This will make the IgG inactive. It cannot do anything. It cannot block phagocytosis, opsonization. It can't do anything, okay. So if you are asked the virulence factors, you should write all. Antigen are virulence, causing virulence. Toxins are causing virulence. Enzymes also causing virulence, okay. So under virulence, what and all you'll cover? Antigen, toxin and enzymes. What will you cover under antigens? For Staphylococcus, Capsule, Peptidoglycan, Teichoic acid, Protein A. Under Capsule, what and all you'll write? Polysaccharide. It will resist phagocytosis. It makes it more virulent. Peptidoglycan, you will say that it is also a polysaccharide. Then it is also a part of the cell wall. It evokes the cytokine response. Okay, then it activates complement and evokes production of inflammatory cytokines. Teichoic acid also a part of the cell wall. You will say teichoic acid always goes in hand with peptidoglycan. Teichoic acid in Staphylococcus aureus will be ribitol. In Staphylococcus others, it will be epidermidis, it will be glycerol teichoic acid. Then again the same thing, it will protect it from opsonization and all that. One second. One more thing, that it will facilitate the adhesion. So these two words you should not forget for teichoic acid. Adhesion, protects from opsonization, okay. Then coming to protein A, antiphagocytic, anticomplementary, and protein A main thing is it will bind to the Fc portion of the IgG and it will block phagocytosis, okay.